What we're embarking on is the nervous system. So it's one of the systems that we talked about that is responsible for maintaining homeostasis. So it's a critical system to your body. And so what we're going to do is we're going to start with the cellular level and talk about how cells that make up the nervous system are constructed and kind of how they work and interact together. And then after we do that, we're going to then look at the grosser overview of the nervous system in terms of the brain, the function of the brain, spinal cord, nerves coming off of the brain and spinal cord. And then kind of end the quarter looking at some specialized sensory apparatus we have. So we're going to look at the eyes and the ears in detail. And then talk about the autonomic nervous system, which is actually the critical role in homeostasis. So that's kind of the approach we're going to take. So today's lecture is actually going to look at kind of an overview of how the nervous system is constructed and then how cells that make up the nervous system interact. On Monday's lab, we'll actually look at tissues of the nervous system and kind of refine what we've talked about today in terms of your visual interpretation of it. And then what's kind of nice with the breaks we have is I'll actually be ahead of lab with the lecture for a change because we have another lab lecture on Tuesday and no lab until the following Monday. So there are three basic functions that our nervous system provides us. So to do homeostasis, our nervous system has to know what's going on externally to your body, so around your body, what's going on internally in your body. And so what we have is a sensory function. And the sensory function provides information toward the central nervous system. So the way we can think about the nervous system is that we have a brain, and then we have a spinal cord that make up the CNS, which is the central nervous system. And it is our interpretive center that takes sensory information. So what we actually have to have is we have to know what's going on internally and externally on the body surface. And then that information has to be conveyed toward the central nervous system. So sensory information moves toward the central nervous system. And then what we say is it's afferent information because it's being conveyed toward the central nervous system. So within the CNS, the brain or spinal cord, we have neurons that receive the impulses and make determinations about the impulses. And so that's the integrative function of that the central nervous system plays. So it takes this sensory input and essentially makes decisions regarding our response to try to maintain homeostasis. And so the neurons involved in that are called interneurons or association neurons. So the one thing that we can see from that is that the cells that make up the central nervous system are called neurons. So that's going to be the name for our cell, the cells that we're going to refer to. Okay. So we have sensory neurons here. And we have inner neurons within the central nervous system. And then what happens is we have to send a signal back to the body. And so the signal back to the body is moving away from the central nervous system. So we say it is efferent. And then because it's going to affect cardiac muscle, skeletal muscle, glands, we say it is motor. So the neurons that carry that information are called motor neurons. Okay. So we kind of create a loop where sensory information comes in via sensory neurons. Inner neurons in the central nervous system interpret, make decisions about the incoming impulses, and then send signals back via motor neurons. So if we put that into more of a graphic picture, then what we have to have is we have to have some type of sensory receptor that is sending signals to your brain. So in the last unit, we talked about a couple of unique sensory receptors that respond to changes in tension on tendons and skeletal muscle. So muscle, spindle apparatus, tendon Golgi apparatus. And that's how you're aware of the relative position of your arms and legs at any point in time. There's a relative tension created. That information is sent into the central nervous system. And then we actually create a decision based upon that tension and then can respond. So one of the things we do clinically to figure out whether somebody's nervous system is functioning correctly is we do reflexes. So if we think about reflexes, the most common one that is done clinically is a patellar reflex. And so remember the quads, the rectus femoris, the vastus, three vastus muscles, all insert on the patella via the tendon of quadriceps. And then the patellar ligament connects the patella to the tibial tuberosity. So what we actually do when we do a patellar reflex is we strike the patellar ligament with the reflex hammer, and that stretches it. And so the tendon Golgi apparatus that are in the patellar reflex then send a signal to the brain. And what the signal is like is you're starting to fall backwards because the patellar reflex is being stretched. So that information would be initiated on that sensory receptor, the tendon Golgi apparatus in this instance. And then the nerve that would carry it into the central nervous system is a sensory nerve or afferent nerve, which is the blue nerve. Now, the way it works is that all sensory information that enters the spinal cord all enters on the posterior or dorsal aspect of the spinal cord. So now no matter what level of the spinal cord you were looking at, the sensory neurons always enter posterior. And then reflexes can be very simple or very complex. And so the one up here we would refer to as a spinal reflex because it's not dependent on you having a functioning brain. So if you have somebody with head trauma, there's a series of reflexes that you can go through. Some of them are cranial reflexes that require a functioning brain. Some of them are spinal reflexes that actually don't require a brain at all. And so if somebody has significant head trauma, a lot of the cranial reflexes will be gone, but the spinal reflexes will still be present. Okay. So the example here is just simply a spinal reflex. So what happens is the sensory neuron interacts with an interneuron or an association neuron, which is the purple neuron up here. And it's the part of the central nervous system that would do the interpretation. So in our simplest reflex, we don't even have an association neuron. And then as the reflexes become more complicated, we can have a number of association neurons. So that pattern changes based upon the sensations that we're referring to. Okay. Then what actually happens then is the association neuron interacts with a motor neuron. And the same pattern exists for motor functions. They all exit the spinal cord via the anterior or ventral aspect of the spinal cord. So motor always enters dorsally. Sensory always enters dorsally. Motor always exits ventrally. Okay. And then what happens is the motor neuron, which is our efferent neuron, goes to an effector. So remember, efferent and effector are connected together. And then the effector could be cardiac muscle, skeletal muscle. If we go back to the patellar reflex, then the patellar reflex would be a reflex where we don't have an association neuron, where the sensory neuron simply connects to the motor neuron. And And then once the tendon is stretched, because the sensation is you're falling backwards, then you contract your quads to bring yourself back in line. That's why your leg goes out in a patellar reflex. And the motor neuron is going to skeletal muscle, the quads, and makes them contract. That's just an example of a reflex. So we're going to do this in a little more detail in Lab 3 when we actually look at the spinal cord itself and how it's constructed. Okay. So there are a couple of words up here that we need to think about. So the brain and spinal cord belong to the central nervous system, which we can use the acronym CNS. So notice the nerves here are labeled as peripheral nerves. So what we would say then is that nerves belong to the PNS, or the peripheral nervous system. And the association neurons belong to the CNS. Okay. So the way that we communicate with our central nervous system communicates with our body is we have 12 nerves that come directly off the brain and largely go to the anterior aspect of your head, your face, neck, and upper shoulders, with the exception of one really weird cranial nerve that goes down into the viscera, internal organs of your abdomen and thoracic cavity. And so we have 12 pairs of cranial nerves that come off the brain directly. And so we're going to begin to talk about those next week on Tuesday when we begin to talk about the brain. And then we have 31 pairs of spinal nerves that come off of the spinal cord and go out to our body. So our total investment in the PNS is 43 nerves. And we're bilaterally symmetric, so we have a nerve on the right side and we have a nerve on the left side. 12 pairs of cranial nerves, 31 pairs of spinal nerves. So perfectly bilateral.
43 pairs.
Pardon?
43 pairs of nerves.
Correct. So we're bilaterally symmetric, so we have 43 pairs of nerves. And so as we talk about this, we're going to put this together in relationship to that. Okay. So there's a couple of other words I want to introduce. So one of them up there is a ganglia. So if we look at the word ganglia, if we just shortened it to gang, if I said we have a gang, what would you think? A group. So ganglia is a reference to a group. So a ganglia is a group of neuron cell bodies in the PNS. So if we have any neuron cell bodies outside the central nervous system, they're aggregated into groups that we call a ganglia. We're going to look at the last slide and look at one in a second. So there's another word. There's another word up here that we've used a number of times. So it's the word nucleus. So if you took a chemistry class, you would interpret the nucleus as the center of an atom. And it still contains a group of entities, right? So in chemistry, what we would find in the nucleus would be
neurons and
protons and neutrons. If I use the word nucleus as it applies to a cell, then we think of it as the center of a cell, typically, but we already know that some cells don't follow that pattern. But what's in the nucleus? DNA. So it's an aggregate of genetic material, DNA. So if we have a group of neuron cell bodies in the CNS, then we call it a nucleus. So English must be fun to learn, right? Nucleus with 3 different definitions for the same word. But the common theme is it's a group of something, right? Okay. All right, so if we go back to the last picture, on this picture, the way that we can describe a neuron is notice the neuron has this bulb at the end and then this long thing that goes out. So up there, if you look at all of them, they've done the same, similar, except for the sensory neuron is a little different. So the bulb represents the cell body. And this represents what we could commonly call a nerve fiber. And in the nerve fiber is going to be one of 2 types that we're going to talk about later. So the nerve fiber is just kind of a generic thing. Okay. So notice that the blue neuron, the sensory neuron, the cell body is outside the central nervous system. The ball, but the ball for the purple neuron and the ball for the red neuron are within the central nervous system. So then where the blue ball is would be called a ganglia or nucleus. It would be called a ganglion, correct. So where the blue ball is right here, we would call it ganglia. And then it's going to have a more specific name that's tied to some structures within the spinal cord itself. So the way nerves enter and exit the spinal cord are called roots. So this is a root, and this is a root. So since this is associated with this, and it's on the back of the spinal cord, then the root on the back of the spinal cord would be called the dorsal root. And the root on the front of the spinal cord would be called the ventral root. So then this becomes the dorsal root ganglion. So it's just descriptive. It's a ganglia associated with the dorsal root. And that would be a very common ganglia. So we have 31 pairs of spinal nerves coming off the spinal cord. So we have 31 pairs of dorsal root ganglion. Okay? So to kind of summarize kind of what we've been talking about and begin to kind of compartmentalize different functions of the nervous system. Then if we're dealing with things arising from skin, muscle, bone, sensory receptors associated with skin, muscle, bone, then we use the word somatic. And so the neurons carrying information off skin, muscle, bone that are somatic sensory neurons. So we would have these somatic sensory neurons that send signals to our brain and spinal cord. They interpret it. And so if it's going to skeletal muscle, then we call it a somatic motor neuron. So that's a voluntary system. So that's one that you create cognition to and awareness of, and you respond to it. Okay? So to make sense out of that, if you have some of those new nice glass top stoves that glow red when they're on, but when they're off they don't glow at all. So let's say one of your family members was using the stove, you didn't know they'd been using it, and then you put your hand on it, and it's really hot. Then you stimulate a sensory receptor in your finger that sends a signal into your spinal cord, and it goes to your brain. And cognition is awareness of sensations. So the amazing thing about that is even though you would say, "Ow, I burnt my finger, it really hurts," your finger doesn't hurt at all. All you did with your finger was stimulate a neuron, a sensory receptor that sent a signal to your brain, and your brain says, "Your finger hurts, you couldn't move it." And then you move your finger because it hurts. So when people have amputations, if a nerve is stimulated at the amputee point, their finger hurts, even though the finger is no longer there. Because it wasn't the finger really hurting. A sensory receptor in the finger sent a signal to the brain, and the brain interpreted the signal and projected the pain back to tell you that your finger hurt. Okay. And then what happens is, because your finger is burning, then you're going to send a signal via a motor neuron to the appropriate muscles that are going to withdraw your finger real quickly. And it's very, very fast response in terms of sensory input, cognition, and motor response that causes you to remove your finger. So we use that strategy. For example, we can do surgery on a hand, and we don't have to put somebody to sleep if we do a nerve block. So when we do a nerve block, we stop the transmission of the impulse to the brain. Therefore, the brain has nothing to interpret and tell you that your finger hurts while we're doing the surgery. And they can do the surgery and you don't hurt. That's the way to think about it. How can we use nerve blocks to do surgery? Because we're stopping the transmission and the brain has nothing to interpret. So the other thing they do on some procedures, like coloscopes and stuff, is they give you a drug that prevents you from creating memory while you're in the procedure. And so then when the procedure's done, you don't remember any pain because you didn't make any memories during the procedure. So we can either do it by playing with the brain or actually preventing impulses from being carried to where they can be interpreted. Right? So if we're dealing with internal organs that we don't want to spend time thinking about, like our heart or our lungs, our digestive system, we don't have to think all day, "Oh, I just ate, I better move something out of my stomach into my small intestine. Oh, now my small intestine is getting full, so I need to move it into my large intestine." We don't have to think about that at all, because our brain does that at an unconscious level. And so we call that the autonomic nervous system, or the ANS, versus the somatic nervous system, which is the SNS. Okay. So the autonomic nervous system does things for internal organs that we don't want to have to think about all day. I mean, just think, if you had to think to make your heart beat every second, if you have 60 beats per minute, what if you got really interested in a movie and your brain got distracted? Then your heart wouldn't work. So we have this autonomic nervous system that takes care of that. It sends signals to our brain and spinal cord, and then the signals back go to autonomic motor neurons. And those are typically go to smooth muscle and cardiac muscle, the two types of muscle that we said were involuntary. And then glands, adipose tissue, and stuff like that. Okay. So your digestive system has its own little nervous system to take care of it. So since the digestive system's inside of you, then it's enteric. And so that is our ENS, which is our enteric nervous system. So those would be enteric sensory receptors and enteric sensory neurons. It can actually send signals to your brain and spinal cord, or it can kind of take care of itself. Or some activity. So it can directly interact with enteric motor neurons. So that one's kind of odd. We're actually not going to do much with this one until we get to the digestive system. So we're going to spend most of our time in this unit looking at this and looking at that. So the other thing that we did, and it comes from the fact that if you actually look at fresh nervous tissue like a brain or a spinal cord, that part of it looks really pale and white, and other parts of it look kind of dark. And so what they traditionally did in early anatomy was just simply call the white stuff white matter, and the dark stuff gray matter. So the brain and spinal cord were divided into white matter and gray matter. And the pattern is a little different. So in the spinal cord, the dark stuff is on the center of the spinal cord, towards the inside, and it kind of forms a butterfly-shaped structure within the spinal cord, followed surrounded outside by this lighter material that we call white matter. And then the brain has all the gray matter on the surface, and then the white matter is internally. So the organization of gray matter and white matter is slightly different in the brain and the spinal cord. But what we find, so the thing you have to remember is gray matter and white matter arose before microscopes, because that's something you could see if you had killed an animal and you removed its brain or spinal cord. And then once we created microscopes where we could investigate tissues, then we could figure out, well, why in the world is white matter white and gray matter gray? And as it turns out, is gray matter contains neuron cell bodies. So if the neuron cell bodies are aggregated into groups, then gray matter contains ganglia or nucleus. Nucleus, yeah. So gray matter is a collection of nuclei within the central nervous system. White matter contains cell extensions, or these nerve fibers. So what we're really looking at in the central nervous system is this would be found as a component of gray matter, and this would be found as a component of white matter. So, and it's just the appearance that it has. So, in the Midwest, people eat a lot more brains than they do in Washington. So, in the Midwest, you can usually go to the grocery store and find brains in the meat market at the grocery store. So it's always fun to go buy a couple of tubs of cow brains and bring them to lab so you guys could actually see what the brain really looks like in terms of this white matter and gray matter. So what we're going to discover is that when we look at a nerve fiber and we try to cross section so we can see it, that we actually have the nerve fiber at the center. And then it's going to be surrounded by a layer of fat. So to make, if we thought of a wire in your house, then the wire is what conducts electricity. So the nerve fiber is what conducts nerve impulses. And since we don't want wires in our house interacting with each other, because they tend to create fires, then we insulate the wires, right? So we put rubber around them or plastic. Well, we don't have rubber and plastic as an entity in us, unless it's been inserted. So we use what for insulation? Adipose, yeah. So this layer of adipose tissue around nerve fibers acts as an insulator and isolates one nerve fiber from the other nerve fibers. Well, if you open a can of Crisco, what color is it? If it's not the butterfly, it is white. So now you know why white matter is white, because it contains a lot of lipids. So if you go to the Midwest, where they eat brains, so I was going through Indiana once, and I stopped at this little, sometimes I get bored with the interstate, so I get off on small roads if I have a little extra time. So I stopped at this little diner, and the lunch special was brain sandwich. And I was like, I've never had a brain sandwich. So they sliced the cow's brain and they bread it and fried it. Well, it was just like fried butter almost when you took a bite of it, because it was really soft because it was mostly fat in terms of the myelin. So it's kind of like melted cheese or butter. Very interesting. One bite was enough. My grandparents had a farm, so they killed animals, and they always ate the brains. So it was always, once you got a little older, you learned the tradition. Is the family would get together, butcher cows, spread the meat out, but you have organ meat, and that's what's ate for several days after you did it. So then you wanted to avoid those days. So people were like organ meat. What was going on? So scrambled eggs and brains is what my grandmother used to make for breakfast. Brains and scrambled eggs and mixing them together. Some people love it. So the other thing, if you think about it, we can have head colds that make our nose run, our eyes water. We can have them in our trachea that make us produce lots of mucus and spit them. So what would happen if those same viruses affected your brain, that affect the linings of your nose and other things? You'd be in serious trouble. So what we need to do is, since our nervous system plays such an incredible role of maintaining homeostasis, then we need to be able to isolate our brain and spinal cord from the rest of the body in a way that protects it from most common pathogens that can wreak havoc on the rest of our body. So what our brain and spinal cord are surrounded by are 3 layers of connective tissue that we collectively call meninges. And if they're inflamed around your spinal cord, you would have spinal meningitis. And if they're inflamed around your brain, we would have cerebral meningitis. So meningitis is an irritation of these layers of the spinal cord, and not surprisingly, spinal meningitis and cerebral meningitis are very, very serious diseases. They can lead to death. Okay? So the outer layer is the thickest, densest layer. So I always like to think about where words come from, because as a student, it always helped me try to remember words. So the outer layer is called the Dura Mater. So then it's Latin. So if you look up Dura in Latin, it's tough, and you look up Mater, and it's mother. So the literal interpretation is Dura Mater is tough mother. So you can envision early anatomists doing a dissection of a cow or a goat, probably a goat, and they're trying to cut through this, and it's really tough with these crude knives. So this guy says, "This is one tough mother." The guy taking notes writes, "Dura Mater." Ever since it's been known as the tough mother. Well, its real role is to protect your brain from being exposed to things that could lead to early death. Okay? So the Dura Mater is this really thick layer of connective tissue we see right here in this picture of the spinal cord itself. Now, just inside of the Dura Mater, we actually have a layer that has a lot of fibers that extend off of it. And so it's called the Arachnoid Mater. So does anybody know what the first part of this word, arach, is a reference to? Spiders, yeah. So they must have seen some daddy long legs or something. They thought these long little fibers look like spider legs. And so it got its name, the Arachnoid Mater, because of these fibers that come off of it. And so this picture is not very good because actually they actually put their verbiage over it. But you can see these little fibers, gray branches in here. And those are why that inner one. So the Dura Mater would be outside, and on the inside here would be the Arachnoid Mater. And then we have one that's directly over our spinal cord and brain itself, right on the surface. And so it's called a Pia Mater. And so the Pia Mater would be just on the surface here. So we have a Dura Mater to the outside, very durable. Arachnoid and Pia Maters, which are not as thick and not as durable. And in between the Arachnoid Mater and the Pia Mater, we actually have a space. And so this space between the purple and the blue is called the Subarachnoid Space. And a subarachnoid space contains a fluid that we make called cerebral spinal fluid. And that actually allows your brain to be, and spinal cord to be buoyant in this fluid, which protects your brain and spinal cord from mechanical trauma and shock. So to knock someone out, what you have to do is to hit them hard enough so that the brain, which is normally protected from impacting the inside of your skull by this fluid, to be moved so quickly that the brain slams on the inside of the skull. And that's why people pass out. And then the trauma to the brain created by that is now called a concussion. Yeah. And so I don't know if you've been paying attention to media, but there's been a lot of concern about concussions in sports, particularly football. And so the amazing thing is, we've been playing football for over 100 years, but we only recently have had enough data from former players that have aged to know that there could be long-term consequences. And so what we see is the more trauma the brain has had in terms of concussions, the earlier the onset of dementia in those patients. So we see an increase in brain function in the elderly that we refer to as dementia based upon those concussions. So we actually have this subarachnoid space filled with fluid to prevent our brain and spinal cord from slamming on the inside of the skull or the vertebrae, which creates trauma.
What was that fluid called again?
Cerebral spinal fluid. And what we can do is, if we do have brain dysfunction and we think that somebody may have an infection in their brain or something, we can actually take a long needle and penetrate the Dura Mater and Arachnoid Mater in the lower part of your spinal column between L3 and 4, 4 and 5, and do what we call a procedure called a spinal tap. And then we actually withdraw that liquid, some of the liquid, and that allows us to do both chemical and biological tests on it to test for pathogens and trauma.
That's also where the epidural is below the
So the epidural is actually in a space that's just outside the Dura Mater. So if we put this in context in your spinal column, so remember that we had that neural arch, then we had the body of the vertebrae, and so your spinal cord is actually located inside this neural arch. And just outside the Dura Mater is a space called the Epidural Space, and it's highly vascular. And so an epidural goes into the epidural space. And they use a big bore needle that goes through all the soft tissue until they get to the Dura Mater, and then they use a real fine needle on the inside of that to same way they do a spinal tap. All right, so if we're going to protect our central nervous system with this layer system called the Dura Mater, then we would also want to protect the extensions off of the central nervous system, our 12 pairs of cranial nerves and our 31 pairs of spinal nerves as well, right? And so what we're going to see is that there's a continuum of connecting tissue, and so the outermost layer around the brain and spinal cord we refer to as the Dura Mater. But as we would observe nerves coming off of the central nervous system, they're also encased in a layer of connective tissue that's protective. So the Dura Mater becomes continuous with a layer on the surface of nerve that we call the epineurium. So the good news is that the pattern is going to be the same as muscles. In muscles, we had a epimysium around the outside of the muscle. We had a perimysium around fascicles. And then around each muscle cell, we had an endomysium. So now you got to get rid of the ending mysium and replace it with neurium or neural. Okay. So the entire nerve is going to be encased in the epineurium, which would be continuous with the Dura Mater around the brain and spinal cord. And then subunits of a nerve are going to be called a fascicle. And that allows nerves to branch as you go away from the central nervous system. Nerves are designed so that these fascicles can arise as branches. And so the connective tissue surrounding a fascicle, while it's still internal to a nerve, is the perineurium. So if I take a fascicle out and I look at a nerve fiber, so for our purposes right now, I want you to remove this word axon and just replace it with nerve fiber. We'll come back and talk about why axon was used there. But for right now, we'll just call it a nerve fiber. And then all nerve fibers are going to be surrounded by endoneurium. Okay? And then that protects pathogens from being able to enter the nervous system by entering nerves, because they're still protected by these connective tissue layers. So there are two kind of odd diseases that we get where pathogens can actually get inside of a nerve, and when they get inside of a nerve, they are protected from your immune system. So there's a group of viruses that get into nerves, and they all belong to a group of viruses we call herpes viruses. And so if you have chickenpox, then the virus that causes the blisters on your skin invade nerves underlying your skin. And once they're in the nerves, they are protected from the immune system. You could have had chickenpox at age 3 or 4. You get to your 50s, 60s, or 70s. You are stressed because of another event in your life, another illness or something. And then all of a sudden you get blisters that reappear on your skin. So that secondary is called shingles. Yeah. And it's created by the same virus. And it wasn't that you were re-exposed to the virus. It's the virus was hiding in your nerves. Yeah. So the others are Herpes Simplex 1, which creates cold sores. So, and it follows the same pattern. Creates blisters on the skin, hides in the nerves. Cold sores go away. Sometimes sunlight and other things, overexposure to sun, causes those to come out. You get a head cold or nose cold, and then they come out. That's why they're called cold sores. And because your immune system is busy attacking that other virus, this virus takes the opportunity to come out and cause blisters again. And then Herpes Simplex 2, which is genital herpes, which you get in the genitalia from intercourse. You can get either one, either place, one or two, depending on what you're engaging in. And they all create blisters on the skin. And you never get rid of them.
How come shingles seems to be so much worse than chickenpox?
Well, the pattern that we see even in chickenpox is that the younger you get chickenpox, the less problematic disease is. And the older the first exposure to chickenpox is, the more problematic the disease event is, with higher fevers, a lot more pain associated with the blisters. Okay. Yeah. So when my daughter was really young, I think about 2, we had a family that took care of her during the day, and one of their younger daughters got chicken pox, and they said, "Don't bring her over." And we said, "Oh yes, bring her over." So we actually just took her over and inoculated her with chicken pox. So she had it in two, so it wasn't near as bad as the older girl who lived in the house had. So then when it comes back, it can be really bad. So the most common pattern for shingles is to follow one rib around your torso, and that's because of the pattern of nerves. The ones that get into cranial nerves can be really problematic. If you get shingles in cranial nerves, you can get paralysis of facial muscles for a while, along with the blistering and stuff. So shingles itself, depending on what nerves it arises out of, can be different as well. Okay. So what's actually happening is the little viruses are hanging in here, and then they escape the nervous system. Yeah.
The shingles vaccine or
It's a reminder. So what happens with I'll do it real quick. So what happens if you have a virus or a bacteria? It's got little identifiers on it. And the way your body knows that it doesn't belong to you is these identifiers. Right? So if you have the appropriate type of white blood cell that comes in contact with it, the white blood cell creates something we call an antibody. Once the antibodies are produced, they bind to these identifiers. Okay. And then once the antibody is bound to the identifiers, then another white blood cell knows it's supposed to destroy this entity. So what normally happens, like you get a head cold, is you've got a virus. Your body at first doesn't recognize the virus, and then it takes a little while for the virus to be recognized. And in the meantime, the virus has been taking over the cell machinery of your cells and producing thousands of copies of the virus. So you get worse and worse, and then you finally get better. So first thing is you have to have this recognition. Then you have to create an antibody. And then the antibody, and we were not sure how it happens, there's a memory for that antibody. So that if we're exposed again, we don't have to go through the recognition phase. Pardon?
Is it Herpes cell?
Yeah. Yeah. So, but even within the cell, we're not sure how the memory is retained. Yeah, which was my point. So anyway, what we know is that for some viruses, we don't retain the memory very long at all. And then for other viruses, we do. So the whole idea of vaccines is to take the virus and the viral code and deactivate the virus with heat or other mechanisms so it can't make you ill, and then inject that dead code into your body so that you go through the recognition phase. And then once you go through the recognition phase, then you'll know how to build antibodies so that the disease doesn't progress as quickly for as long a period of time. So if you think about common flus, you have to get a flu shot every year because we don't retain that antibody very well. Tetanus every 5 to 10 years, depending on who you talk to. But others we seem to retain for a long time, like hepatitis. It appears that we do that. But if you had a hepatitis vaccine and you're exposed to hepatitis, you could actually go to the doctor and have them do an antibody analysis to see if you are still producing antibodies. And if you're not, then they can do a booster, which will bring your antibody production back up. So there are some vaccines we need repeated boosters for. There are other vaccines that, as far as we know, we don't. So a classic example would be smallpox. We used vaccines to totally eradicate smallpox, almost worldwide. I'll bet a lot of you in here have never had a smallpox vaccine because it was eradicated. It was eradicated by the time you were born, so you didn't need it. Us old folks, we have a little scar on our arm where the smallpox vaccine was given. So, pardon?
Military has it stuff.
Yeah, military get it because there are all these vials of smallpox viruses in laboratories all over the world. And so if somebody actually were to get their hands on a vial and use that as an agent to make people sick, then there would be a large part of the population that's unprotected because they don't have the vaccine. Now we actually did that in our early history when we were coming west and having conflicts with Native Americans. We sent blankets to Native Americans, that people who had smallpox and been in contact with, and it decimated large populations of Native Americans with the disease. So that's kind of the way the virus, the antibodies actually work. All right. So when we look at neuron, then what we can do with a neuron is we can create kind of a pattern, and then once we have a basic pattern, then we can look at differences in different types of neurons, and it'll begin to make sense. So we have this entity where the nucleus of the neuron is found. And so it's called the cell body. So we have this entity that we refer to as the cell body that actually contains the nucleus. And one thing we're going to see is that neurons have fairly large nucleuses compared to what we're used to seeing in the cells we've looked at in the past. And because the nucleus takes up more of the cell, it's actually not as densely loaded with chromatin. And so in most of the cells we've looked at in the past, the nucleus stains really dark. But in most neurons, in slides we're going to look at, the nucleus actually doesn't stain as dark. It is quite large. And sometimes what students make a mistake with is they see a really dark staining entity and they think it's the nucleus, but it's actually the nucleolus, which is the area where the DNA that helps us make ribosomal material is isolated in the nucleus. So the nucleus oftentimes doesn't stain very dark, but we get this really dark structure within it that we call the nucleolus. And then the other pattern that's kind of interesting is that we've talked about ER now kind of in three ways. So we talked about smooth ER, rough ER in the first unit. Smooth ER making lipid-based compounds, rough ER making proteins, particularly proteins for export out of the cell. Then we talked about the sarcoplasmic reticulum, which was modified to store calcium. So we have a pattern of ER in neuron cell bodies that makes the cell body look really grainy. And so it's been referred to as missile bodies. So the pattern of ER is referred to as missile bodies. So in lab on Monday, we're going to look at slides of neurons. So one of the things we want to do is make sure that you can identify a cell body. And once you identify a cell body, you can recognize that the nucleus is fairly large, oftentimes doesn't stain very dark, and internal to the nucleus is this dark staining entity, that's the nucleolus. And then the cytoplasm will have this real grainy pattern to it, and that grainy pattern are the missile bodies. Okay. All right. So neurons are designed as communication cells. And so neurons have to communicate over distances. So to put this in context, the neurons that control some of the really small muscles that we didn't learn in your feet, their cell bodies are in the lower part of your spinal cord. And then their nerve fibers become part of a peripheral nerve that go all the way to your foot. So you have neurons who are longer than your leg in length. So because these neurons have to span distances, then they have extensions off the cell body that we call cell extensions. Okay. And so one way we could look at it would be the neuron up there, which is going to have a really long cell extension coming off of it, and then these shorter cell extensions associated with it that we're seeing up here. So this thing and these things. So traditionally what has been done is we've looked at direction of impulse travel. So to compare this to what we've done already, so we did the brain and the spinal cord. And so we said impulses toward the spinal cord are
Afferent.
afferent. Impulses away from the spinal cord are efferent. So now our reference point is going to be the cell body instead of the central nervous system. And so we can have impulses away from the cell body, or we can have impulses toward the cell body. So traditionally, what we've used is the word axon to describe the cell extension that conveys impulses away from the cell body. And then the word dendrite to describe cell extensions that convey impulses toward a cell body. And for most of our knowledge of the central nervous system and the nervous system itself, we have always assumed that neurons have one-way impulse flow. But there's some new evidence that suggests that might have been a bit simplistic. So what we're learning over the next couple of days may change in the next few years. Okay, with some new research that's been done. So if the impulse is traveling away from the cell body, we would call it a axon. And if an impulse is traveling toward the cell body, we would call it a dendrite. So then, all it is a matter of looking at how things look relative to the cell body. So, the example up here, the one I drew, would be a motor neuron. And so most of the motor neurons would be efferent neurons, going out from the central nervous system to your body to interact with skeletal muscle, cardiac muscle, smooth muscle, glands. Okay. So if you recall, when we looked at the other one, and we drew it as a cell body where the cell body wasn't at the end. Okay. And so this was the example in the picture. So if we went back to that picture, this picture. So what the interpretation of this is the cell body's at the end. So it's at the end. So they don't draw the little dendrites, but they draw the axon. So that's these two here. Okay. So notice that this one has this entity, this cell body, and then an entity here. So if we're following the same pattern, if the impulse is going this way and this way, then this would be referred to as a axon. And this would be referred to as a dendrite. We follow that same pattern. Now, we're going to see a little later there could be some confusion with that in textbooks. But it's the same pattern. All right. And then all we're going to see is we can have variations off of that. So we can actually have the cell body in line, and we can have a cell extension on either side. And then if impulse travel is like this, then this would still be a axon, still be a dendrite. So the pattern is, how are the cell extensions organized relative to the cell body, right? And we use the word axon and dendrite to describe that characteristic of the tissue itself. So I'm approaching this somewhat simplistically now, and then we're going to build on it. So we're creating a foundation. So on Monday in lab, what we're going to do is we're going to look at slides of the nervous tissue. And so one of the things we'll see in it are things like this. So this is a cell body. This is the nucleus. And the really dark thing we're seeing is actually the nucleolus. And in this grainy pattern, the cell bodies are cell bodies. And then we have these darn things coming off of it. And it's going to be difficult unless you see a good pattern to it to know actually whether they are axons or dendrites. So if we just call them neurofibers, then we don't have to worry about what direction impulses are traveling. Does that make sense? That's why I use the word neurofiber to begin with. If you knew what type of neuron this was, and you knew that there were many short ones like this, so if we were looking at this neuron, and these are actually short ones, then we would know they are dendrites. If we saw a really long one, we would know that it is an axon. That's hard to do sometimes under a scope.
Are you expecting this to do that, or just verify?
No. Not unless it's something like this. So this one, this is a really large cell extension, which is going to be axon, short cell extensions, dendrites. So this would be a classic mode neuron. And then the other way is if there's context to where the neurons came from. So if the neurons came from the ventral aspect of the spinal cord, we would know they were motor neurons, right? Just from location itself. So again, here's the nucleus. In this instance, the nucleolus didn't stay very dark, but we have this grainy pattern again, which are nissal bodies. All right. So this is just another way you could do it. So you can take a little block of spinal cord, which you collected from an animal. You could put it in a solution of silver nitrate, put electrodes on either end of the block of tissue, run current through the nervous tissue. Then anything that conducts current will plate silver to it. And so this is a cool way to let you see the shape and structure of a neuron, but you can't see the internal stuff anymore, like the nucleus, nucleolus, but you can actually see the cell extensions much better. And so this is just kind of a cool way of doing it. And that's the way they kind of figured this out, was using techniques like that in staining that allowed us to see characteristics a little differently. So we have a cell body, but then we have many different cell extensions again. It'd still be pretty hard to tell because it's functional, not structural, knowing which one was an axon or a dendrite. So we can just call them nerve fibers or cell extensions. Okay. So let's go back to this slide. So the cool thing about maturation is, and newborn babies are so cool, but they're kind of like little blobs. And I say that affectionately. But they don't respond to much, if you think about it. And then about at 6 months, it's like they discover the hand. And then they start playing with toys, and then they just spend a lot of time. And what's happening is they were born with their central nervous system and peripheral nervous system incomplete, and you go through a process of further development. And one of the processes that was incomplete was the process of isolating nerve fibers from one another. And then what happens is we have these cells called Schwann cells. And the Schwann cells actually begin to line up in single file along a nerve fiber. And so each one of these is a Schwann cell. It is actually not part of the neuron. but it's actually a cell that associates with the nerve. And they line up in single file along these nerve fibers. And what happens is there are little junctions where they don't actually cover the nerve fibers. So these junctions are called nodes. And so they're called nodes of Ranvier from the French guy that figured it out. Okay. So nodes of Ranvier. And so these cells are called Schwann cells. And what they do, which is cool, is that while they're doing this, if you're looking in cross-sectional view, here's our nerve fiber. They wrap. And as they wrap, they create a layer of fat that surrounds the nerve fiber. So the green is the fat. And so we call this layer of fat, this yellow layer here, so this layer of fat is called the myelin sheath. So see the myelin sheath is this layering of fat. And so it's an insulator. So one of the things that had to happen as you maturated from that cute little blob to being able to crawl, to being able to hold your head up, to being able to eventually walk, manipulate things with your hands, eventually be potty trained, is you myelinated your nervous system. So then, not surprisingly, there are diseases where you unmyelinate your nervous system. And as you progress through the disease and the myelin disappears, you lose the ability to discretely control skeletal muscle. And you actually take a course backwards toward being that affectionate pig blob. And I don't mean that in any disparaging way, but you lose control of your ability to control skeletal muscle. Eventually you lose the ability to control your urinary sphincters, your anal sphincter, and you go back backwards because you unmyelinated. So those diseases are hideous because they just reverse the process that you went through in development.
So, will you still be able to sense whether or not something is touching your pinky or your thumb, or?
No, eventually you lose almost all sensations.
Okay.
So like with MS, if you're demyelinating, what is there not a way to like inject someone else's Schwann cell and have it?
There's been some research on that. Yeah, there's been some research on it. The first thing, and the other research is trying to find where in your genome the issue lies. And so we could approach it from a genetic repair, or we could try to approach it from a different mechanism. The problem with taking someone else's Schwann cells and injecting them is if the disease manifestation is destroying Schwann cells, you'll just destroy the new Schwann cells. They're just playing there unless your Schwann cells. Yeah, unless your Schwann cells are having a unique characteristic to them. So that's a couple of things we'd have to look at to resolve that issue.
Is there a specific name for the disease?
Pardon?
Is there a specific name for the disease?
Yes. And there's a couple of others.
MS is the problem.
Yeah, that's the
Spokane
case.
loves it.
Pardon?
Spokane loves MS.
Well, what's interesting is that MS appears to be related to distance from the equator or closer to the poles. And so there's some people that think there may be a magnetic response to it. But if we look at individuals born in more northern latitudes compared to individuals born near the equator, MS occurs in greater frequency the closer to the poles you get and further away from the equator. But now that we can see that pattern, we still have to interpret what the pattern is really about. Right? Is it just a chance effect, or is there actually something correlated with it? But that is what we know not only Spokane, but if you just look at major metropolitan areas like Detroit and kind of in this same latitude, that NMS would be higher in people born there than it would be, say, people in Dallas
Is it
in that same latitude.
Students Spokane with the highest concentration in the lower government?
Yeah, we do have a fairly high concentration. And what we refer to that in genetics as a founder effect, just because of a chance occurrence that people who located in Spokane apparently had a greater genetic affinity for England, so it elevates it more population. And then that could change as more people move in and out. So one thing we're going to do on Monday is we're going to look at a slide of a nerve. So this is a nerve that's been cut. Each one of these is a nerve fiber. And this is actually the nerve fiber itself. This white stuff is myelin. So now, completing a thought process for us, we said that white matter in the central nervous system was white because it was high in lipids. Myelin is the entity that's high in lipids. So white matter is white in the central nervous system because it's dominated by myelin. Okay? Right. So this is a nerve fiber. This is the myelin. And then this is a junction between one swan cell and a neighboring swan cell. So this depressed area here is a node. So it's a node of Ranvier. So if somebody is better at French than I am. All right. So to think about what we were talking about, so if we're going to look at cell bodies, and we're going to look at how these cell extensions arise, then what we can do is categorize neurons into different groups based upon configurations, right? And so the pattern for most motor neurons is they are multipolar neurons because they have many short dendrites and one long axon. And then connecting this picture to something we talked about last time, if we had a skeletal muscle cell attached to each one of these axon terminals, we would have what entity? Well, these would be the intermuscular junction. So what would that entity be? A motor unit. So remember, a motor unit was a motor neuron and all of the cells that it interacts with. So if we had a skeletal muscle cell with each one of these terminal axons, then that would represent a motor unit. And that's actually how a motor unit is constructed, because you have one multipolar neuron with all of these axon terminals. So if we go to this one, then this is called a unipolar neuron because the axon or dendrite is actually coming into the cell body at one point, so there's only one pole. And then this is a classic example of what type of neuron we've already talked about. Sensory neuron. So the two we've talked about already would be this multipolar neuron, or classically what motor neurons are. And this unipolar neuron is classically what sensory neurons are.
So when you use unipolar neuron, why is it named the neuron fiber axon as it's going to the dendrite?
Remember I told you there would be some confusion. Yeah. If you want, for our purposes now, you can just cross axon out by dendrite. And then that'll help you remember that if it's being conducted toward the cell body, it would be a dendrite. And then we'll pick up on that later. Okay. All right. So this one's kind of cool because you have this entity going into the cell body and this entity exiting the cell body. So impulses toward the cell body, dendrite. Impulses away from the cell body, axon. And the cell body is in the middle. And so we call this a bipolar neuron. And bipolar neurons are very important integrating neurons in the central nervous system. Where we're going to see one on the slide is actually in the retina of your eye. So the retina of your eye is actually develops from an outpocketing of your brain, embryologically. So the retina of your eye that collects light and creates our visual field that we have is actually part of the brain, and it has it has bipolar neurons. So we'll actually be able to see bipolar neurons when we look at a cross section of the retina of the eye.
Does dendrite not myelinate, or is that just a picture?
No, that's one of the reasons why this was labeled as an axon, because there are some people that interpret myelinated as being an axon and unmyelinated as being a dendrite. But that just clouds the issue, which is probably all going to change because it's not as uniformly one way as we thought anyway. So. The beauty of science is the more we learn, the more we understand, and oftentimes we have to go back and correct what we had interpreted wrong, right? And it's not that science is bad, it's just that science is doing its job, yes. All right, so these are pretty cool. Purkinje cells and pyramidal cells, where they have this cell is designed so it can interact with thousands of other neurons simultaneously. Yeah. So Purkinje cells are found in the brain. So one place we would see Purkinje cells is in the brain in an area where we store memory. And when you connect neurons together in linear pathways is what we call a memory trace. So all quarter long, when you've been learning new stuff, you've been making new neurons interact with each other. And as long as those neurons continue to interact, you retain the memory. But if you have depth to some of those neurons, like in a stroke event, then the memory is gone. So in Alzheimer's, Purkinje cells die. And as Purkinje cells die, they continually lose the ability to make new memory. And they continually live in a world of old memory as long as those Purkinje fibers are still good. And as more Purkinje fibers die, their memory begins to fade more and more and more.
Is that why you do long-term old stuff that you can't remember?
Yeah, exactly. And it happens in dementia as well, in various types of dementia. So both my stepfather and my mother had dementia. So I've watched the progress over time. So I brought them here a number of years ago so I could take care of them because they couldn't manage for themselves. And then they reached a point where it was beyond my capacity to manage that, so I put them in the care center because their memory just kept fading and fading. So my stepfather doesn't recognize me at all. Doesn't. I can walk into the room, he has no clue as to who I am from anybody else. And my mother just has recently got to the point where she's a teenager. So she doesn't remember me at all because she's too young to have had me. So it's a very interesting progression to see cells like these Purkinje cells die. What we know with Alzheimer's is there's a gene for a protein, and a protein begins to be produced and it covers the cell. And as the cells get covered by this protein, they become functional. So the long-term hope for Alzheimer's is we can unravel why that gene is making the proteins and figure out how to stop the proteins from covering the cells.
So do the cells die, or do they just release?
They die, actually.
They die.
Yeah, you can actually. I compared an early MRI of my stepfather's brain to one recently, and in an area of the brain called the hippocampus where long-term memory is stored, he's got a big hole in it. There's actually a space that shouldn't be there. So the atoms have actually died. So it makes it an unreversible disease that eventually kills you. And the present drugs we use slow the progression of the disease down, but don't eliminate the progression of the disease.
Does it typically affect the newest memories first?
Pardon?
Does it typically affect the newest memories first?
Yes. Alzheimer's. Yeah. Yeah, new memories they get. And then they get into these repeated patterns. So when my stepfather was still home, and my mother would go somewhere, if I was over there to kind of make sure he was okay, in an hour, he would probably ask me 70 times where my mother's going. It would just be this repeated question. He'd tell him, he'd go away a little bit, then he'd come back and ask me, because he really couldn't remember.
Do you know if there's any reason why it's sex in those ones first, or is it just?
It's the fact that when you make a new memory trace, you have to make neurons interact. Since you got a degradation of neurons, there's less neurons to actually interact.